plants, or it cannot be done in the microbes or anything. So that's where it starts, the importance of the biotechnology, how to deal with these kind of issues. And once they find out the routes, how to do the production, or how to make it as a drug in a alternative resources, then they will have to find out how much efficacy or potency is there in this molecule on the patient. Then they need to check that initially, that they will be done through preclinical studies by doing some of the animal experiments. They cannot directly do it on the humans, because we need to know whether the product what you have been trying to find out, like example insulin, whether it is purified in a good way, whether the target dosage is good enough to give the efficacy, potency, or the desired effect what you are looking for. To see all of that, whatever you have done in the early discovery, you need to find out its impact in the preclinical studies. That can be first checked either in the animals or some of the tissues which are isolated from the animal or animals itself. Once if we see and identify this, this has good amount of the efficacy and potency and all, then it will go to the clinical phase. These are very crucial. Phase 1, phase 2, phase 3 clinical stages will be there, and each and every phase is different, and what they are looking for, and the data that is getting generated is more important. I personally myself know in the place where I'm working, Ipsen, there is a drug which they found out that has really good. They took around 10 to 12 years. It has good efficacy, potency, everything is fine. Preclinical studies were fine. Phase 1 and phase 2 studies were really good. They got all the results what they want. But when they moved to phase 3 recently, the dosage is not enough. It was found like that because the in the phase 3 clinical trials, they the trial of human number is more compared to the previous stages. Now we came back with some queries from the USFDA. We are addressing them. It's not necessary that whatever you found out in the basic research or early discovery, whatever the great results you found, it's not necessary that it will make a good drug at the end of the day. If you see out of 10 molecules which were 10,000 molecules which were successful with basic research, probably for 5 or 6 are really going to be into the market. Even after that, we are not sure. After there are molecules, drug molecules which were released into the market, but because of some other side effects or issues which were not identified through the phase 3 or phase 4 clinical uh, trials, they have to recall it back, and they have to completely close that off. There were issues, so it's always going to be very critical. It's a money taking process. That's why if you see whatever the drug discovery, it is not generally done by any of the government bodies. They do, but it's all up to the major basic research or early discovery. But beyond that, they won't go because it is going to cost, especially at the clinical stages and all. Yeah, like I said, if we start with 10,000 compounds at the basic research levels, then by the time you see preclinical compounds, it will come down, narrow down to the 250 compounds only. By the time you reach clinical trials, it will become just 5 compounds. And FDA will review, not sure out of 5 how many will get approved, probably one or maybe less. And if you see the amount of time it takes for all this process is around 13 to 15 years, which is really huge amount of time. And for a young guy like him, it's a lifetime experience if he involved in all these processes. So what are the major steps, especially in terms of biopharma, if you see, what are the major steps involved in the drug discovery process? First, we need to identify what area, what kind of disease they are targeting, because each and every organization will have their own priorities. Like some they work on cancer. Even in the cancer, there are hundreds of different types of cancerous effects. So what kind of target they are looking? Is it something related to bone, or is it something related to heart, or is it something related to blood? Once if they decide that, and what in that, they need to find out what is exactly the target identification. Even in the particular disease, what are the impacts of the disease on the human body? What are the symptoms they have been observing? They need to find out all of that. Then they need to understand, okay, this has this particular symptoms, and this has this effect on the blood flow, or effect on the organs, or negative impact. Especially I'm talking about the negative impacts. When they come to know that these are the things, this all can be done only through the testing which can confirm the symptoms, or impact or damage of the organs. That will give more idea what kind of molecules to be targeted later. So the first thing is we should be able to identify our target in a way that we need to do a lot of research on the target itself. It's not even the drug. First we need to find the characterize the target, characterize in terms of every level, whatever it is possible. In biopharma they use lot of technique: cellular and genetic targets, genomics they do, proteomics they do, bioinformatics. Bioinformatics you must have heard, proteomics and genomics are also you must have heard. But apart from this latest, they are using artificial intelligence. We'll come to that point once we discuss about all the basic steps that are involved in this. All these steps you must have seen in your academics and all. Uh, One thing, if you want to ask or raise a question, you can just interrupt me and ask me, no problem. Once we identify the target, the what is its characteristics, then we can think about the molecules. What kind of molecules will be suitable for this? And how do we find them? What kind of molecules we need to screen them? And what kind of testing is needed to test these, to try these molecules, biomolecules against the target? All this can be done through the synthesis and isolation, which is the very primary step in the lead identification. Then you find a certain group of molecules which are suitable. Then you extract them, purify them, and then you try to find out the interactions between the target and the drug what you identified. It's just basic research, but optimization of this interaction will go into the next stage. But for doing even this basic research of the interaction of drug molecule or hit molecule or lead molecule with the target, you need to have certain essays also. So at this stage itself you will be having some basic essay development, which without analysis you cannot do anything. You know the molecule, you know the target, but without analysis you are not going to do anything, because you should find out whether the desired therapeutic effect what you are trying to see. How do you confirm that? It is through some analytical method developments only. Whether it can be the simple HPLC or SDS page, or it can be very very effective, like the somatic cells or stem cells in the biotechnology. But you need to do the testing at the stage itself.
Sir, sorry to
So
interrupt you, sir. Sir,
yeah,
can you please play the slideshow, like full screen?
full screen.
Yes, sir. Now it's okay.
Okay, sorry. Okay.
Okay,
Yeah,
sir.
once we identify target and find out the lead is not going to be one single molecule. So it is like hundreds of molecules. You see that at the lab level, hundreds of molecules will have very similar impact on the target, or else the diseased tissue or whatever it is. You'll think that all are they're going to work definitely, but that is not going to happen in the next stages. When you when these two are identified, then you will have to find out the optimization of this drug interaction with the diseased target or diseased tissue or whatever it is. Now, how do you optimize the interaction between them? How any interaction can happen? First of all, it is through there will be a certain site on the drug molecule which is very specifically going and binding to the target tissue. It can be through the protein-protein interaction. It can be through the cell membrane to the protein the interaction, or it can receptor especially, or it can be through the enzyme and all are related at this level, or gene level, or yeah, protein and gene levels. It happens in the biopharma. So how do you optimize them? You must have heard that there will be analog molecules or modification of the molecules itself. So how do they modify? There are different techniques. Adding particular molecule which can stabilize the interaction, or prolong the interaction of the drug with the target area, or you can do some chemical reaction for the protein molecule so that its the structure gets little bit modified, which is still 99% similar to the original one. All of these modification will lead to the more impact and more efficacy and more potency of the drug to act on the target tissue. So there are different studies, search studies in silico screening and also chemical synthesis. If you see, I'm talking more towards the biopharma. I know you all are having pharmacy background, but it is nearly the same. When I say chemical synthesis, exactly it is involved the chemicals, but they
Excuse
do
me, sir.
prepare the peptides. Yeah, please.
అది కొంచెం స్లైడ్ మోడ్లో ఉంది కానీ షేర్ చేసి ఫుల్ స్క్రీన్లో పెట్టరా ప్రజెంటేషన్ మోడ్లో.
Think it is still in the. I'm seeing. Are you seeing this small
Yes,
side
yes.
one? Okay.
Yeah. Small one we are seeing.
Probably I should. Actually, I have two screens.
Sir,
One of the screen, I'm seeing the big
yeah.
one. I don't see this part, so that's why I couldn't
Please,
find out.
please go to display settings, sir.
Display.
On the top
Where
of
I get.
the screen, you have display settings. Just above the slide, sir. Just above the slide, you have display settings.
I don't see that.
On the top of your screen, sir, you have show taskbar. Beside it, you have display settings. Yeah, that is show taskbar. You have display settings just beside.
Next one.
So this is not
Just the next.
seen on my screen. That's the reason
Oh, okay, fine, sir.
I.
You do one thing, sir. Please end the slideshow. End the slideshow.
Where exactly is that? Really?
Yeah, on the top again, or you can just press escape.
This page is not happening. Sorry. I'll stop the video. Sorry, even if I close the video, the slideshow will not. Yeah.
Yeah. Yeah, you can press this. Yeah, that one. Yeah. No, the same thing is coming.
Oh, okay, carry on. Now share it, yeah, full screen, yeah.
So that option is available in the display settings. I think you are not able to see the display settings.
Yeah,
right, sir?
this is it got stuck behind this mute option for me.
Okay, okay, fine. Hide presenter view.
I'm trying. Yeah, yeah.
Yeah.
Thank
Yeah,
you.
okay, thank you.
Thank you. Thanks for your patience. Yeah. Okay. Where we are? The optimization of the lead. There are studies in the drug discovery which can modify the drug molecule and increase the impact of the drug on the target tissue. So yeah, SAR studies, in silico screening, these are uh, you know SAR studies are kind of old kind of studies. You check them in the lab with direct molecules with the drug. This in silico and chemical synthesis are kind of bit advanced. Yeah, but in terms of pharma, chemical synthesis is quite old. Like each and every time there will be solid support. They add one amino acid and they block the some of the sites in the amino acid. They add the second amino acid and then third amino acid. It's a trial and method. It's kind of old method only, but this all the data is being used nowadays to produce the drug in high quantities using the data analysis and all. So this we can say in silico screening is also uh, used latestly, even though it is not too latest. Once we do all of this, we find we optimize. We know that the drug is able to provide definitely the target uh, dosage which is needed and all at this level. But how do we confirm that? Uh, how do we find? How do we further give clarity that it is easy from this stage onwards it can get into the clinical trials. So for that we need to do the preclinical studies. This first it involves in vitro studies and then in vivo studies. What is this in vitro studies? In vitro, as you all know, this is in the lab. We try to it very much similar to some of the activities of the in vitro studies are closely relative to the lead optimization, where you try to improve the affinity. But this again it comes into the in vitro studies also. Not much difference would be there. The purity or the amount of screening you are doing at this level would be very high, because by the time the lead optimization is done, the assays what you are using, still you need to validate them. Once you validate at this stage, lead optimization stage, then that validated methods only you can use when you reach by the time of preclinical studies. Up to then it is completely, you say, R&D. It's a process, kind of process R&D, which involves the the interaction of the drug with the target tissue. But once you enter into the preclinical stages, by the time you will have a lot of data which you are going to completely talk about the drug efficacy, potency, how it is what is its nearly shelf life and how long it is taking and what kind of impact it is there on the animals, or this will be clearly can be seen and ratified only in this stage. But before that you cannot get that. You could see only some of the lab experimental data. Since we are able to find that affinity is good, selectivity is great enough, and it is really showing working on some of the cell disease models. Cell disease models are nothing but you create a disease in the system itself, then you try to infuse the drug. This is how you try to find out. and then you see mechanism of action. You have to go to the level of the molecular pathway, how it is impacting. Of course, by this time you would be doing some background work through all the previous stages, but the mechanism of action has to be clearly explained here at the molecular level itself, so that it give more confidence for the any reviewers who will approve this to go into the clinical trials. Then lead candidate refinement is also, it is the same, nothing but uh, optimization again. So these are very closely related, but you try to do on some of the cells or target tissues here, whereas in case of lead optimization, still you do the thing, but there is a minor difference. And in vivo studies at this point of time, instead of working in the lab, you try to use the animals, like how it is shown in the especially mouse, most of the places in the biopharma. But nowadays there are issues with using the animal models, so they're trying going towards the microbial models majorly. In this in vivo studies, they try to see what kind of animal models they can select, and what are the disease states in the animals itself. How do they behave with the disease? How do they behave when they get infused with the drug? How can they do the functional imaging? Like functional imagining is nothing but you see the mirror of one molecule on the another one, and find out how it is going to impact them. And ex vivo studies, it's nothing but doing instead of doing them in the direct animal itself, they take some tissue organ and literally work on the tissue organ. In the previous stages, when I mentioned about the tissue or organ, they don't do them directly on the tissue. They do extract the molecules or genes or proteins, whatever they are, behind the cause of the disease. That is how they do target there at that level. But here they infuse the drug and they extract the tissue, and they see after few days of drug effect on the organ, then they will do the studies on it. Once we cross all of this, we get the approval of the preclinical, then we will be going to the clinical trials, where it called phase 1, phase 2, phase 3 are major. But nowadays there is phase 0 also. I'm not sure how many of you heard, but in industries, by the FDA, they have been asking about the phase 0. What is the difference between all of them? Phase 0 is nothing but very close to phase 1. But the thing is, there will be certain amount of drug dosage. It has to be given to the patient, right? By the time of preclinical studies is done, it is completely, standardized at that point. When they decide that, for example, zero point 0.5 mg of this protein is needed for the patient to infuse to work on the disease in single dose. But in phase 0 study, they don't give that zero point 0.5 mg per ml of the protein dose. What they do is it's exploratorily they increase like 0.1, 0.2, 0.3, 0.4 on healthy volunteers, and they check at these different doses, which is lesser than needed dose, how it is impacting them on healthy volunteers. It is asked recently by US FDA. And then anyhow, we know phase 1, phase 2, phase 3 will be there. Phase 1 is again on the, probably some numbers in tens on healthy volunteers. It's not going to be on people who are affected by the diseases. And they see only majorly the concern is with respect to the side effects, because they don't have the disease. So once if they are given the drug without the disease, first thing what do they see is side effects. What are the side effects? How it is impacting them over the period of time, how it is impacting their food habits, how it is impacting their behavioral aspects. All this will be recorded in that. Then in phase 2, they do try the drug on actually people who got affected by the disease, but the number of people will be less. It's like, around 100 to 200 or 250. It depends. The size of the population, what they choose. Again, in the phase three, they do on like, more than 300 to 1,000 people. And there is phase four also, but generally that is termed as post-marketing vigilance, pharmacovigilance in general. They say, well, what is that phase for? So it is being given into the market, the drug is being given into the market, but what happens? Job is not done. That's where the major responsibility comes from. Because so far, what we have done is it's the number of people we tried is it's a small size, nothing in terms of the whole world population. When it enters the market, it is going to get into the crores of the people's body. So the impact and what is happening, because each and every human individual is different, right? So how they are responding for each and if we see in UK, what happens is the medical care is run by the government completely. There is nowhere private organization can take care of the medical care. So, for example, if I go to the hospital, what happened? What kind of things they have observed in me? What did they report them? Everything gets noted into the system where it provides the data can be provided for many number of years whenever it is needed. So that is more important, because if anyone tried with some new drug, all those implications of the new drug will be recorded there, that will be extracted and used. But in some places, in some countries, it is not possible, because it's a situation based medicine. They give it and they get it done, and it got cured off, and no record of what is happened with them. In the larger perspective, is up to clinical trials, it is fine. But once if it is enters into the post market, the data availability is quite less. So it is difficult to uh, you know find the root causes, what happened, where it happened, and what kind of patient he is. Is it male or female? What was his another health situations? So this situation is called phase four, actually, pharmacovigilance. Coming to the target selection, as I said previously, first of all, what disease it is, what damage it is there in the tissue, and how what kind of molecular targets are involved in the disease. Can you see the screen? It's going now. Okay. And once they identify all of that, target validation will be there. How do they do the target validation? How it is what kind of tests, what kind of the tests, or what kind of molecules they get involved in that. There are a lot of bioactive molecules, cell based models, protein interactions, signaling pathways. Have you heard of signaling pathways? Especially it gets deal in biotechnology. The signaling pathways are like, if you see in our body, or sometimes when we go to hospitals, they do check some level of sodium, potassium, all these things, or they do check how do they get maintained, that sodium or potassium. This is just one example I'm talking about. So through the membranes, through the cellular membranes in human body, the flow of sodium or potassium or calcium, whatever, it happens through the membranes. Then there will be certain concentrations they have to be maintained through this membrane pathways, or signaling that is happening in the membranes. If that is not happening, then they will see the different variations of concentrations in our blood levels. So this is what. And another example of the signaling pathway is like, I'll talk about the insulin. How does insulin controls the glucose? It it doesn't go and directly involve in the glucose control. It controls the loop mechanism, where it go and controls the some of the ATP molecules, and then it gets the directed towards the glucose metabolism. That is how it gets controlled. It is not that insulin is going and binding with the glucose or something else directly. These the whatever the interactions that are happening in between the control of the glucose and with the insulin, these are called as signaling pathways. And functional analysis of the genes. How do they find out the functional analysis of the genes? You must have heard about the PCR, polymerase chain reactions. First, they try to isolate the genes or genomes from the targets, and then they see in what way this diseased gene is or diseased genome is different from the actual healthy genome. So they try to image both of the genome and find out which are bit abnormal, and the abnormal genes which is majorly towards responsible for the disease. In that way they find out. It takes really a lot of time, but nowadays, because of the last 60 70 years of the research work on the human genome or any other plant genomes or bacterial genomes, the data is available immediately, and to compare, there are softwares to compare them. It is possible nowadays. That's the reason few diseases they got the drug very in the very short span of time. I would say example COVID-19. That's really short span of time. And the data that was utilized, there is one drug called remdesivir, like remdesivir, yeah. The drug for COVID-19 is it was not meant for COVID-19. It was meant for some other viral diseases. But the data that was generated by this usage of the drug in the market, that has given the clues that can be used for the COVID-19. Then they started doing some initial studies on that, and they found it was really good. That is how this, the latest technologies or genomics or protein interactions or cell based or the data, all of them are able to give us immediate results. But for all of them, we need to have the basic research. All the genomics, that the genomics is nothing but like human genome. What are the genomes? What are the different genes are there in the human genome or plant genome? What is the responsibility of each and every gene? What kind of enzyme or what kind of protein it is translating into, what kind of protein, what kind of enzyme, how they are impacting, in which signaling pathways they are working, what is protein-protein interaction, what is the receptor-protein interaction. All this can be started first of all with the Sanger whole genome, which was quite old technique actually. Nowadays they are doing whole genome sequencing itself very easily, and these can identify what gene or what protein is responsible, and whether it is possible to target at the gene level or is it possible to target at the protein level. Before I talk about the gene and protein, it's better I give clarify about, the genes are there. Genes, they get converted into the genes are nothing but DNAs. DNAs gets converted into the RNAs. RNAs gets converted into the protein. This is how the process happens. So which level it is better to target? Is it at protein level? Is it at RNA level, ribonucleic acids, or is it at a DNA level, which is the primary source, or deoxyribonucleic acids? It depends. It depends on how you identify the target, how you see, how do you characterize that. Depends on that. Whether the interactions are strong at protein level or whether interactions are at strong at the gene level. How that depends. What is the triggering the disease, and all this characterization will be able to done through this genomic and proteomics. Proteomics, yeah, yeah, identification of target receptor enzymes, whereas some biological approaches for the focus is at the gene or transcription level. Transcription is nothing but converting the gene into the ribonucleic acid, as I said just before, DNA, RNA, and protein. DNA gets converted into RNA. This is called transcription. So whenever they target the identification, whenever they see the target identification at this genes level, it is especially cellular and genetic targets, these are called. And whenever they go to the next level, that is called protein interactions, where we discuss them in the proteomics. Drugs usually act on either cellular or genetic chemicals in the body, known as targets, which are believed to be associated with the disease. This point is very important. Believed to be associated with the disease. Then how do we know that they are believed to be associated with the disease? Like I said previously, the whole genomic studies, the cellular and genetic targets and genomics are nowadays combined together. This next one and this one, because in the initial days they couldn't do the whole genome sequencing in very short span of time. So that's why what they do is they used to target the a specific tissue genetic material only. So in those days, that's why they targeted only the gene and transcription level. But but if you see nowadays, they are using the genomics, where it is easy to find out about 35,000 to 50,000 genes in the human genome very easily. It contains nearly 3 billion nucleotides. Imagine 3 billion molecules means interacted molecules are there in the body, and finding them through the just lab methods, how it is going to be, how long it is going to take. It was done in the old techniques previously. Now they are using that into the latest advanced methods using artificial intelligence, machine learning, and all. They are using this genomics and finding out the targets and how what exactly their behavior, and how the disease is impacting them, and what symptoms it is showing, what kind of impact it has on the blood flow. All these will be uh, recorded with respect to that particular gene. Then it is easy to give the target for that particular gene. The number of genes implicated in disease, both those due to defects in single genes and those arising from combinations of genes, is around 1,000. So out of 35 to 50,000 genes, there are around 1,000 genes which could, give the diseases situation in the human body. And imagine there are 5 to 10 linked proteins per gene. 5 to 10. One gene is not, it's not that one gene gives one protein, but to give that protein, there are another proteins get involved in that gene. So there are nearly 5 to 10 linked proteins per gene. The number of potential drug targets may lie between 5,000 to 10,000. Like 1,000 genes are there which are responsible for the diseases, and each and every gene is linked with 5 or 10 proteins. Then 10 into 5 or 10 into 10, it is going to be around 5,000 targets or 10,000 targets are there. One of the best method they use is single nucleotide polymorphism. They compare the genomes from the both healthy and sick people. That is what I was talking about. This kind of imaging both the good gene and sickle people gene, and identify where exactly these genomes vary, and wherever it gets vary, the target that gene or the molecules involved in the gene pathways. Coming to the proteomics, this name itself says the proteome. It's a study of the proteins. This is the third level where DNA gets transcribed into RNA. RNA gets translated into the protein. So this third stage, and if you see most of the targets in the biopharma industry is at the proteomics level. Most of the drugs, whatever they identified, they are probably 70 to 80%. They are at the proteomics level, because sometimes the genes you know might get mutated, and they try to behave a little different, but at the end of the day they are going to produce the proteins which are 99% similar to the protein what it was previously. That's why it is easy or good to target at the protein levels. And one more thing, one example I would like to give, HIV is a viral, right? previously there was no medicine. It is difficult. It is nothing can do anything. Nothing can do for this disease and all. That's because viral genome, it gets mutated each and every time on its own and change its way of behaving. Then if it is changing like that, it is quite difficult to target that kind of diseases. That was the reason. But by this time there are a lot of drugs for this, because there were a lot of basic research happened once this disease is identified, this kind of disease is identified. And through the latest technology, just by seeing the data, they would say there is a possibility if we use this particular drug, it can, it can uh, give some potential results which are very much positive towards this disease. That is how this genomics and proteomics together nowadays they are working. In this proteomics, they do see the protein-protein interaction and how the protein is going to interact with the nucleic acid, how the protein is going to interact with the ligands. All these are within the cell material, inside the cell or on the cell, these interactions happen. Yeah, as I said, target identification in the proteomics is performed by comparing the protein expression levels in normal and diseased tissues. It's the same as how it happened in the genomics. Like, they compare the healthy genes with the, diseased gene. In the same way, proteomics also do the, healthy diseased healthy protein expression and diseased tissue protein expression. They compare them and they see how the characteristics are changing. How can we target that molecule? This is what totally proteomics talking about. And in this, it's not only 2D page. There are a lot of techniques like isolation of the protein. It starts with isolation of the proteins, and they do the reduced SDS page, normal SDS page. These are all talks about the LCMS. They talks about the molecular weights of the protein molecules. They talk about the structure of the protein molecule and how it is the what whether it is polar side is getting exposed or non-polar side of the protein is getting exposed, which part is getting interacted more towards the target tissue or diseased tissue. All this will be characterized fully using the many techniques in the biopharma industries. Bioinformatics, I would say nowadays bioinformatics has become a bit old, but still it has good amount of data that can be used in the latest methods of methods which are using the data mining. It's a branch of molecular biology where the molecular biology is nothing but which talks about the genes, RNAs, and proteins. So this is all called molecular biology. And also it talks about the data using of the biological data using in the computers. Once in previously when the bioinformatics was introduced, it was really a big revolution. Similarly, how the artificial intelligence nowadays it is doing. It was easy to identify the target, and there are some chemical compounds which can be easily screened using this bioinformatics to find out the drug and to give some whatever genomics and proteomic interactions we talked previously. It can be done through the bioinformatics as well. That is how it advanced. But the thing is, it needs software and all. This can compare the entire genome of the pathogenic or non-pathogenic strains of microbe and identify genes and proteins associated with the pathogen. If you see Clostridium botulinum, it's a toxin-producing molecule which generally happens in the food poisoning, but it is a highly toxic that we can die within in the less span of time. So the whole genome was it's a pathogenic, so the whole genome was studied previously, and they had comparison with different variants of other pathogenic genomes and they identified there are certain proteins that are being released by this Clostridium botulinum has severe impact compared to the other pathogenic toxin molecules. We are actually working on that Clostridium botulinum here. It produces Botox. Actually, the name is called as Botox, but still it is converted into as a drug in our company, and they are using it for some medicinal purposes. Yes, well, now we saw there are drug diseased tissues. We identified them, and we understood the mechanism of disease, what it is doing to the body or to the tissue. Now, what exactly the next step is? Lead identification. Lead is nothing but the drug identification, or sometimes it is called the hit identification. What molecule is needed to target the previous work where we identified all the mechanism of the disease. Identification of small molecule modulators of protein function. Especially, you see, even though you talk about the genes, at the end of the day, it comes to the protein level. So yeah, all the identification is going towards the protein molecules. Whether you see antibodies for some cancerous antibodies are nothing but proteins. If you see the toxin molecule, what we are producing here, then that is also a protein molecule. If someone they say we are producing streptokinase, which can be used for the blood clotting issues, that is also enzyme. Enzymes are nothing but proteins. So if you see many of the drugs, even insulin is a protein. They say hormone, but it is protein first of all. Then it is classified as a hormone. So 90% or minimum 75% of the all the drug molecules are proteins only. Process of transforming these into high content lead series. Lead identification involves a lot of things. First of all, we need to find out which group of drug molecules are the molecules have very high potential to work towards the diseased target. So when they find out these are the certain group of molecules who they can show that caliber, then they try to separate the mixtures using some of the extraction purification technique, and then or else they can do the target selection itself. They find out that this kind of molecule is needed to target, or this kind of structure should be there for the drug molecule to target this disease. Then according to that, they design the molecule, and they synthesize that molecule, or some of the biosynthetic intermediates can be used directly after the minor purification levels. So how do they do that apart from the just synthesis and isolation? Isolation is like, it's a it's not completely purified one. It's you just take the tissue of the drug like plant extract. You use some solvents or some buffer, or you take some microbes which can produce the drug molecules like penicillin and all. You crush them, and you use the buffer, extract it into the buffer where it has the stability. Then you take that solution, site on the drug molecule. This is what this isolation is maximum to that level only. And but there will be next level of purification where they purifies in such a way that all the other impurities, when you extract something from some tissue, it's not only the drug molecule that is getting extracted. Other impurities, other enzymes, other proteins that are being in the microbe or plant tissue, they also get co-extracted into the solution. They Apart from the drug molecule, everything is impurity. It might be useful for the microbe, it might be useful for the plant, but apart from the our drug molecule, everything else is impurity. So we need to purify to the extent such that it has 99% of our molecule, or probably very close to 99.9% sometimes. Then there is another way of identifying the lead, high throughput skin. Once they isolate these molecules, they do take the solution from the target, like the target disease target, they do extract the enzymes or proteins that are involved in this cause of disease. They take that solution. There will be big plates where they can drop 5 ml or 1 ml or less than 1 ml solutions into that, and they can put different types of these drug molecules, like thousands at a time they can run. They can take those 1,000 drug molecules or potential drug molecules onto this target disease tissue at a time and find out which is showing the best results of acting on that disease target. That is what called high throughput screening. At a time they can screen a lot of molecules to see whether they really have the potential to reduce the disease or not. And one more thing that is needed, more importantly, is assay development. You take the drug disease drug target, and you take the uh, uh, as you're thinking that this molecule might be having definitely drug potentiality to reduce this disease, and you combine them. This is up to what happened. But now, once the reaction between the drug and the tissue happened, how do you identify it? You need to have certain assays. So in biotechnology, I see in pharma A plus B is gives C and some byproducts, and then you see the, suppose if C is the product. If I'm wrong, please don't mind about the pharma equation. That you see the activity of that C on some chemical assays and all. But in biopharma, what they do is it's all biological it is biological reactions majorly, so it is going to be very complex. They do test this, but when they take the uh, reaction, when they isolate that reaction whatever drug plus a target mixture compound, they need to find out how the interaction and the mechanism happened. Like enzyme, suppose if enzyme is the drug, an enzyme is the drug, how it enzyme needs substrate interacted with, and how much time is taking, like how much efficiency is there. This is how they do the assay development, and this is very initial level of assay development. In the later stages, they do have to optimize that. But to find out this interactions very clearly, everyone needs this assays. In the biopharma, it can be especially molecular biology technique. Then they do it through some cell-based assays. They do it through some tissue-based assays. They do it through just to find out SDS reduced SDS page. There are different types of assays like this. And what is this combinatorial screening? So either they rapidly synthesis through the chemical reactions, or they try to do the computer simulation to find those structurally related molecules and search the new leads and optimization of the target. This is where it starts with the next lead. After it is almost near the end of the lead identification, optimization of the target affinity and selectivity. This involves some modification of the structures and ADME, then they'll see ADME properties through these some of the assays and studies, absorption, desorption, metabolism, and excretion. But nowadays there is one more term called ADME. I think it is K. K means by the end of the presentation, I'll try to recollect that. There is one more, the ADMET, I think. Yeah, it's a toxin. Yeah, it's not only talks about what what the drug has done to the body. It not only talks about what the body has done to the drug. It has to talk about the toxicity. I'm sorry. I think I need to find out this point again. It is also important because In terms of academics, when you see, we always talk about ADME only, but there is a new terminology recently introduced into this. This is nothing but pharmacokinetics and pharmacodynamics. You all must be knowing about this. It talks about how the body is getting affected by the drug, and how the drug is, uh, transported across the different places, and how it is impacting the diseased area, and how it got metabolized. See, the drug doesn't live in the body. It has to remove from the body. So the metabolism gets involved there, and it takes the drug molecule through the different pathways, and then it gets excreted through the I think most of the times it is urine. So there is one more property. I'll find out that. Yeah. So this is the lead optimization. Like I said previously, they do modify the molecule. So how do they modify? How exactly they can do that? When they find certain molecule is having the potential to act as a drug on the particular disease, then they'll be starting looking into the that protein and the drug and protein interactions. How strong the interaction is, that strong, and how long it can, how effective it is. So when they see that through the some of the SAR studies, so these are site specific related studies, and they need to find out what part of the drug is slowing that interaction. That is called actually pharmacophore. How they need to identify that through the studies, and then the pharmacophore is that is the precise section of the molecules responsible for the biological activity. And if you try to modify this molecule a little bit, it can show more activity than what it was original. I'll tell you one example of insulin. Insulin is some 50 amino acids, or I think 50 or 55 around amino acids are there in that. If they change only two particular amino acids in the insulin, that will become glargine, insulin glargine molecule. But insulin has very less half-life in the human body compared to that. Just changing in one or two amino acids in the whole protein sequence is increasing the half-life to 4 times nearly, from 6 hours to 24 hours. In old days, people, I remember, I think they used to take insulin twice a day or thrice a day, I think so. But there are nowadays insulin analogs. These are actually called as analogs. A minor modification will be there. These are called analogs, and they give more shelf life. They do have more shelf life in the human body, and their impact on acting on the disease is for a long time. That means these analogs are getting more attention than the original molecules nowadays. That is how these in silico screenings or studies are quite really important. Based on these studies, then they do the chemical synthesis of the molecules. If they are very small molecules like insulin also produced through the chemical synthesis, but not all the biological drugs can be produced. Because, for example, if you see the monoclonal antibodies, MAPS, like GCSF, granulocyte colony stimulating factor, that is also one protein, but it's a very like 100 or 110 amino acids would be there in that, or maybe more than that. These are called very large proteins. You cannot synthesize them. It's very complex. So you cannot block so many sites to attach one molecule and one amino acid to the another amino acid. It's really difficult. Yeah, making some 50 amino acid synthesis is going to be a bit easier. That's why some companies they produce the insulin molecule which is chemically synthesized, not through the uh, normal natural process. Once they alter this the molecule, the shelf life, as I said, it is. So there are different ways of altering this, either doing at the genome level, Like just now I talked about the insulin and insulin glargine. Insulin glargine is the analog molecule of insulin. Insulin gene, if they change two amino acids which are different in the insulin gene at the gene level, not at the protein level, if they change, then it will be able to produce the molecule closely related to insulin. So that change happens at the gene level. That is called rDNA technology, recombinant DNA technology. And coming to the same insulin example, this is produced only in human. Then how the companies are able to produce the insulin molecule apart from the chemical synthesis? I know I told about they do it through the chemical synthesis, but there are ways to do it through the microbial or biological process. What they do is they synthesize the gene that is responsible for the production of the insulin protein or insulin hormone. That gene, they'll inject that into the E. coli bacteria. This will be done through the recombinant technology, or this is called rDNA technology. Once this gene is injected into the E. coli genome, that gene gets accepted by that E. coli genome and it gets expressed. Expressed means it produces the protein. Here, whatever we injected is responsible for the production of the insulin. So the E. coli, if you provide some food, like media, food means in terms of energy sources for the bacteria, it grow, then it will produce the molecule. Its own molecules also gets produced. Along with that, our target molecule called insulin also gets produced. Now, instead of the insulin gene, if we change a gene for one particular amino acid in that insulin gene, it produces the analog molecule. But the procedure will be the same. Only thing is an amino acid will be little different from the insulin molecule. So this improves this all will be done through the understanding of the genomics, understanding of the proteomics, and obviously the basic lab techniques like rDNA technology and all. Once if they do that, then they'll be able to produce the larger quantities of the drug. So similarly, this in silico screening and SAR studies will be able to optimize the interaction of the drug disease target and drug molecule by modifying its structure, by finding out its affinity. Then the half-life of the drug increases and its impact also increases on the disease place. Yeah, just now I was talking about what kind of examples are there. How do they do this, all this affinity, selectivity, or how do they do that? The examples are they derive the cells from the multicellular organisms, starting with E. coli to mammalian cell lines, Chinese hamster ovary cell lines, which produce the monoclonal antibodies. These are multicellular organisms, but E. coli all are the single cell. And there will be subcellular components like ribosomes, mitochondria. I hope you have come understanding of all the biotechnology. Um, and then they'll be able to use the extracts of the cellulars. They do try to crush the cells using high pressures, and then they extract whether it can be DNA, RNA, or proteins. These these will be tested. Once they extract, these will be tested in the lab to find out the drug affinity and selectivity. These in vitro studies will be based on the lead optimization or medicinal chemistry. What we got some data in the previous stage, and yeah. So how exactly drug affinity is the ability of the drug to bind its biological target. It can be cell membrane. Each and every cell has membrane and it has receptors for the transportation, or receptors for the signaling pathways, or receptors for the binding of the proteins or enzymes, etc. These are all the places that gets targeted. Then once they find that it has to specifically, what is the specificity? It's nothing but specific. Selectivity is nothing but the specificity. It has to have that specific binding nature, otherwise it can go and bind to the any of the target, so it shouldn't happen. Example, aspirin. You get a pain in one of your body parts, you take the aspirin, but how it is going and exactly acting there itself, that actually the when it is in pain, or when it is in some diseased condition, it releases some molecules in response, in output. Those molecules or those enzymes or those outputs are getting targeted by the drug, whether it can be aspirin or whatever suitable for the pain or disease. This is called specificity. It go all through the way in the body, but it doesn't bind in another place, in another places or another cells, but it goes and bind in the particular place, and that is called selectivity. That is how the lead optimization, or there where you will find out some level of mechanism, and that will be proved in this in vitro studies. Then isogenic human disease models. These are all family of cells that are selected or engineered to accurately model the genetics of a specific patient population. Sorry, just hold on. Sorry. Yeah. So this is what the genes have to be compared and modified at these disease models. And another one is stem cell disease models. This is very advanced technologies. Adult or embryonic stem cells carrying or induced to carry defective genes are investigated and to understand the molecular mechanisms and disease characteristics. Stem cells are nothing but they isolate an organ, and uh, those cells related to the growth of that organ, or stem cells. And another thing is mechanism of action. So you think that yes, it is going and bind directly to that particular place only. It has that affinity. The drug has that selectivity. But how do you know? How do you see that? How in what way you prove that? This is called mechanism of action. Qualify a drug, or to find out how it is going to inhibit the target and meet the other criteria: solubility, permeability, protein binding, selectivity, metabolism, toxicity profiles. These are all involved in the mechanism of action studies. And optimizing lead hits for clinical trial is commonly referred to as lead optimization, which was there in the previous stage. This refinement in the structure is necessary. So optimization is very much necessary. The drugs, they do have the selectivity, they do have the affinity, but they might not have that potency. They might not have that strong selectivity. They might not be able to give the required impact for a long time. So this will be done only through the optimization. And once you do that, then the structure will be able to the potency, safety, and also sometimes improved selectivity. Because why I say that improved selectivity is that there are targets, disease targets, or normal targets. They might have very minor differences in terms of structure, very minor differences, like 5% only difference, but the remaining all the other structural similarities are there. Then the drug if that the selectivity is not strong enough, then it might go and bind to the closely related places as well, or cells as well, or membranes as well, or receptors as well. So that this modification or lead optimization, it can be improved through this some of the in vitro studies by modifying them and finding them. What are the in vivo studies? In vitro studies are nothing but the lab based studies which is useful to find out the potency and find out the mechanisms of the actions which can improve further. But once if you get a lot of data from them, then how you are going to prove them these are having that activity, and we cannot do them directly on the human beings. So we'll have to do some in vivo studies using some of the animal models and find out what is their metabolic profile, because we have seen some in vitro studies showing some properties of the metabolic properties and drug interactions and some toxic nature through the assays what they were developed previously. But how do you prove them in the live bodies? That's the reason they select the animal models. What they do is, in the animal models or in the in vivo studies, first category is animal models of disease state. They induce the disease or injury to the similar to the human conditions. They take the healthy babies of the mouse or whatever it is. They take them, they induce the diseased conditions because they already studied that how that condition came to the human beings, what is responsible for that, or what protein or what enzyme, what interactions are responsible for that. So by knowing that mechanisms and all, they create the same situation in this animal body, and that mechanism of the cause must be equivalent, very much equivalent to the, like, if they get attacked by the virus, or the similar situation has to be created for the virus. It's not that directly injecting or something, that they create the environment so that it get exposed to that, so which makes it very much equivalent to the mechanism of the cause. And it will be able to predict the human toxicity in 71% of the cases. That is what the data says. What exactly that means? When the human is getting in, struck by any disease, he will show some symptoms, or some kind of bad situations from his body, right? So the similar kind of observations also can be seen when the animal models got exposed to this disease conditions. So most of the times, both human model and animal model have seen similarly. They match, the symptoms have matched 71%. So if we see the examples, they used CID mice in case of HIV, NOD mice in diabetes, and January. These are different varieties of mice, because it's almost very similar, but they are given the names because of the geography and small variations in the genome and their behavior, etc. And there will be behavioral studies which needed to be checked in this in vivo studies, like when the animals given the uh, certain amount of dose of the drug, they start behaving because of the impact of the drug differently. Like they might lose the weight little bit, or they might gain the weight, or they may walk very fast than the previous before it gets injected, or they eat a lot of food. uh, Sometimes they sleep more. These are all comes under the behavioral studies. Many of the things will be recorded, but there will be some which cannot be identified. Definitely self-esteem and suicidability are very hard to find out them, but they can see the depression, because you can literally see the it's it's not sleeping, but eyes are open, and a couple of times I have seen the drug induced mice. They become very dull. I mean, which shows some depression kind of situations. There are categories. They note it down and they record them, and they see very excitement type of running. All the corners of the box everywhere, and anxiety. They try to make them forced swimming and tail suspension. So they all will be observed. And in the functional imaging, I think I told previously about this. The changes in the, it's like, finding out, imagine one molecule with the another molecule. This is what in the previous situation I told about the functional. But here exactly, that means how before the drug was induced, the metabolism was how after it was, like how much food is being taken, how the blood flow is, how the some of the absorption capacities of the drug, all this will be studied. They use a lot of tracer molecules for that, to find out the blood flow issues and all. They use the scans, and one more thing is that ex vivo studies. All these previous three were like they literally use the mice models or animal models, but in the last one ex vivo, they take a tissue or they create a tissue in they take that and create an environment which is very much suitable to the outside the organism, and they alter the natural conditions later by subjecting that tissue to the disease. This why nowadays they are doing this. Using some of the mice models is causing ethical issues. It's not easy to get permissions for doing the preclinical studies as well, especially in some of the countries. That's why they're doing ex vivo studies, and examples are measurements of tissue properties, realistic models for surgery. Now we understood that there is a target selection. Once we in the target selection, we see that what kind of disease it is, what are the enzymes or proteins or genes involved in that, and I mean, it's not one particular gene or one protein molecules, because there will be certain number of molecules involved in the cause of the disease, but which is giving there is a degree of cause. Some are 50% responsible, some are 10% responsible. So all that information you will be seeing in the target selection. So then in the lead identification or lead discovery, you will be finding out what kind of molecules are majorly suitable for the particular high degree of target. The methods are done. You identified that molecules, then through the lead optimization or medicinal chemistry, you alter the interaction between the lead or drug molecule and the target tissue or target molecules. You optimize that, you find that this is showing a lot of potency and efficacy towards the clearing of or stopping of the reaction, then it will definitely have that impact. Then in vitro studies, you'll find out that this much uh, variations or interactions, alterations, and in vivo studies, you will see the literally the impact of the altered molecule or synthesized or purified molecule impact on the animal models or ex vivo studies, like where the disease. y- This is so far it's fine. No one is going to object anything except for few animal studies. No one is going to talk anything to you. Everything is fine. But now this is the place where you get a lot of questions, where you get a lot of push to the backside. that is clinical trials. These are procedures in medical research or in drug development. First, whatever you talk, the first thing is how safe it is. They they don't ask how effective it is. The first thing they ask is how safe it is. Then they will go to how effective it is. Because efficacy can be definitely managed or improved through the drug dosages or drug alterations, little bit modifications in the structure and by doing the interaction. But in spite of doing all of this, the first question you get back is how safe it is. So that some safety data you will be giving through this animal studies or in vivo studies, that will give some level of confidence. Then they'll tell you to apply for the phase one. If you don't get that safety data, enough safety data, or enough confidence in the safety of the drug molecule, what you prepared, is going to be really tough. All these clinical trials are essential to get the marketing approval. This might take more than 7 years. It's very in ideal situation it is 7 years, but in the worst cases it can go beyond 10 years. So there are majorly three phases. Phase 4 is a bit different, but actually there are three phases. Nowadays they are talking about phase 0 and phase 4 clinical trials also. Phase 0 is nothing but they don't give the drug dosage what is designed for the disease. They don't give that much. They give less amount, or they try to explorate that concentration into the healthy volunteers. Then they get some safety data. That's why phase 1 is always about the safety. Phase 0 and phase 1 are always about the safety. If you prove that safety in the phase 0 and phase 1, phase 0 is not mandatory, but nowadays USFDA is trying to implement that as well, because a drug dosage of zero point 0.5 mg per ml, for example, 0.5 mg per ml, which is supposed to be given to the deceased person, if you are giving that to the healthy person, the same dosage, we don't know what implications would be there. So what USFDA recent plan is that phase 0, where instead of 0.5 mg per ml of the protein in the drug, we'll start with 0.1, 0.2, 0.3, 0.4. Some criteria will be there even for that. This is just example. Because the dose is the minimum is the impact, like side effects. So if they see the some unwanted side effects at the small doses itself, that's it. No, it's not going to happen. Forget about the phase 1. You need to work on that. So that's the reason. And in phase 1, they give exact the same doses, how much is needed for the diseased person, but to the healthy volunteers, like 10-15 or 10-20 or 10-30, they get the safety data. And in phase 2, they take some 100 people of deceased human, and they try the exact doses on them. Then they see the efficacy and potency characterization, or their how they are, you know, they monitor them. They literally monitor everything, like how they did in the sorry, how they did with the animal models, like weight loss, metabolism, blood changes, and some of the signaling pathways changes. They take the blood samples and check. They record all of the data. And in phase 3, in phase 2, it is a controlled selection, like just 10 to 20. But in phase 3, it is bit uh, vast selection of the people. Like range of the ages also would be more, and the male female age difference is very high. Like that, they do select the large population to try, but the drug dosage and the observations, the recording, the data collection, everything will be almost same as the phase 2. Once if they get, yeah, one point I would like to tell you about the phase 3 is that when any company or any organization, They start the drug is discovered and done. It is all the studies are done. In fact, preclinical phase 1 and phase 2 are also done. But when it comes to phase 3, they cannot do the production of the drug for the phase 3 trials how in the same scale that is being done in the phase 1 and phase 2. If they propose we are going to use the same scale, like for example, in the labs we do in the small test tubes or in the bottles to see all the studies, initial basic research. By the time in phase 1, phase 2, when it comes time we reach phase 1 phase 2, we try to do the fermentation process or this biological complex process in the liters of 10 to 20, 30, 40, 50, like this it will be. By the time the phase 3 phase 2 approval comes, if the company says that yes, we are doing only at 50 liters even in the future to supply the drug to the market, then okay, no problem. You already proved at the phase 2, you are going to do the same at the phase 3, 50-liter capacity only. But there are some other drugs, majority of the drugs in the biopharma, they don't do at 50-liter because it is not, worth doing. It is not, in terms of business, it is not worth. What they do is they take 6,000-liter capacity, 10,000-liter capacity of the fermentation, big vessels. That is where the scale up things come. So when you are trying to do the phase 3 clinical trials, you need to come up with the drug which is produced at the scale what you are intent to do for the market approval, not at the 50 or 10. You want to do in future at 1,000 liters, yeah, do it at 1,000 liters and supply that material to the phase 3 clinical trials. So you get the data back, then you get the approval to release that into the market. Then what is phase 4? Phase 3 is just hundreds of population. They tried the drug on, but in phase 4, phase 4 is nothing but the market data mining, market. It's called pharmacovigilance. The drug entered the market, and it's being taken by huge number of population. Geographically, it is different in like different countries. Like male, female, they do have different other health conditions and all. If someone is very healthy, there is no health issue. They're taking the same medicine. The other person got diabetic and the second disease. They are trying for this drug for the second disease, but they do have the diabetic. So it's all more complicated at this level. And if one single issue, just recall the drug. There is a stamp on this drug that it has some issues. So it's very difficult to get. Once it is getting recalled too badly with comments, then it is really going to tough for the company to handle the situation. That's why pharmacovigilance, the phase 4 is really critical compared to all the previous situations. So they do monitor. Nowadays, they're monitoring so closely for each and every drug in the market, especially in the Europe and the US. They track each and every detail and they get the feedback from each and every place. Even the small doctors, when they prescribe, if they find any incident, even it is minor incident, they are supposed to report it. That is how actually in the place where I am working, Ipsen, it is called Ipsen. So they are doing it. There is a team next to us. They do handle all these queries and find out and record that to use that in the future or to find immediate solutions for that. If you see the expenses of a new drug development, majority of the expenses will go in two places. Around 25% in the discovery place, around 30% in the clinical trials phase. All other things are, less time consuming and but only these two places where they take more time and more money. So far, whatever I talked, you all must be knowing definitely because that is what the pharma or biopharma is having. starting the target identification, lead identification, optimization, some studies to do the preclinical work and then clinical work and post marketing. This is all what happens. But nowadays there is data that is only based on the data they are doing the drug discovery. This data driven science has overtaken traditional lab science, chemistry, and biology as well. A significant majority of the new jobs in pharma industries demand data. This actually, I added this point especially for the young guys who are sitting there, just doing some lab work, uh, or getting experience with some HPLC or some analytical methods, or it's not going to work near future, like 5 years or 6 years of time. Of course, you all guys have to start somewhere, entering into the lab in some industries and all. That is definitely sure. That is this basic understanding needed. But to survive once you enter into this market, it is really going to tough if you are not getting updated as per the technology. This is where the data driven science is playing key role. They wanted to have blend of skills, not just I know pharma, I know these formulas, I do these lab experiments. It's not going to work. I know this biological technique. It's not going. For example, my situation, uh, just before I came to UK, I was working in R&D. There was no data analytical skills, but we means all our departments were, they were really good at doing lab work and all. But how you do generate over the period of time, you do generate lots of data. Just imagine one drug is in the market for the 20 years, how much amount of data would be there from the doctors, from the patients, uh, from other places at all. How do you deal with the data? How do you deal with the data? How are you going to analyze the data? It's not going to be easy. Just see the in the Excel, you cannot conclude anything. Even Excel is got updated nowadays to do the data analysis. Bioinformatics, to some extent, till now it has solved some problems, but it is getting replaced. I think I should say it got replaced by machine learning, deep learning in the artificial intelligence. This is what exactly the data. You see 500 millions of tweets every day. You see 294 billion emails every day. 4 terabytes of data is produced by just a connected car, 4 TB. And this is gigabytes. Facebook, 4, I don't know what is that, P bytes, I think petabytes. 4 PB of data created by Facebook, including 350 million photos, 100 million hours of video watch time. You are being watched every time. Everybody thinks it's not happening, but you are being watched every time. Video calls, lot of data is getting. Similarly, lot of data for biopharma is getting stored. So how exactly, I mean, what way artificial intelligence in pharmaceuticals or biopharmaceuticals drug discovery, it is helping. If you see the previous, I have put this as a, one-way line: target selection, lead identification, lead optimization, preclinical studies, and clinical studies. Now it is a circular now. Target identification, validation, hit or target, lead optimization, all this preclinical and clinical. Again, it goes to the target identification. How exactly this is happening? There are hundreds of closely related molecules. How do you identify the target? sorry, how do you identify the lead? How do you identify the drug? Lead is nothing but drug here, what we are talking. When you start with 10 thousands of the molecules, why do anyone start with 10 or 1,000 molecules, or 100 molecules to identify the drug? Because they all of drug, those 100 molecules are closely related. You see some potentiality in them to reduce this disease condition. That's why you start with 100 or 1,000 molecules. So you have some data of that. You have lots of experimental data that is stored here. And imagine there are hundreds and hundreds of drugs. Just take the case of cancer. When cancer is there, it has 5 or 6 different types of proteins are related to that. There is one drug, but these 5 6 proteins are related to the another proteins, another tissues as well. It's not one protein is working with only one particular gene or tissue or something. It's they do have a lot of connections. Like a single person has hundreds of connections. Similarly, it has hundreds of connections. Then the work what you are doing at the lab level, it gives some inference or conclusion with respect to the similar molecules. That's how they're saying that the clinical data, whatever they are getting, once they identified a drug, the clinical data or the market data, patient data, whatever they are receiving, that will be useful to identify new target and new drug to work on the target. That's why this artificial intelligence is playing key role. I couldn't get one example. Uh, I read it just recently in the news. There was one woman in US. She she got some issue. There are some symptoms. She has been consulting the doctor. Doctor was trying, very similar kind of brands and drugs related to that symptoms and all. But it's not going up. It's kind of going off and on, off and on. She has been really stressed. Then the doctor, what he did is, in US, they store the patient data. What are the everything is stored. So he contacted the company back. This is what the data. This is the issue. She is not getting off. But this drug is intended for all these symptoms only. What's happening? Then they tried all this data, and they checked it in artificial intelligence through the uh, machine learning. They found that there is another drug. It could definitely help us, which is already in the market. It's not that it has to be go through the clinical trials again and again. So they tried that the similar drug, closely similar drug on her, and she was cured off with this condition. This is why it is saying this clinical data again target, and there would be minor differences in the symptoms and situation of the disease and all. Similarly, how there would be minor differences in the different drugs and molecules and all. So this data correlation is giving exact drug. This is called drug precision, no, precise drug identification. 5% of all Google searches are health related. Healthcare data will experience a compound growth of 30% through the 2025, especially in Europe and US. Every time they talk about the healthcare and safety. Only these 2 points. Even starting with a school, the first thing they talk is how safety this issue is, how safe it is, what kind of risk assessment is there, what kind of precautions or preventive things you implemented. So that is how the lot of data is getting generated. And recently FDA it has set goals for big data, clinical trials, and artificial intelligence. Yeah, this is the source where we got. Artificial intelligence holds enormous promise for the future of medicine. That's why FDA also focusing on improving the efficiency of clinical trials, also I mean, drug discovery as well. FDA is increasingly turning to patient-reported outcomes and wearable device data. This is what I was giving the example, patient-reported outcomes. So the data of the patient, what was the disease, what was the symptoms. As far I knew, most of the companies, most of the big shots, like AstraZeneca, Johnson and Johnson, and Ipsen, they spend nearly 20 to 25% on their R&D. And nowadays, we all started using the artificial intelligence. If not to the drug discovery, there are many companies like I work on both the process development for the purification of the drugs and also work on the data analysis of all the manufacturing and also some of the data from the analytical departments. So they are spending so much on data analytical skills and artificial intelligence. We don't have particular knowledgeable person in our company about the artificial intelligence. So recently, a year back, the Ipsen hired machine learning scientists specially to deal with all the data. And now the team has grown to nearly 10 to 15. But see, some of them they do not have pharma background. Some of them they do not have computers background. But it's a blend. It's a blended team. Like some have one expertise, some have another expertise. But I'm suggesting you, guys, if you have both the skills in the next 5 to 6 years, it's going to be a huge revolution of the in the drug discovery, and also it's not only related to drug discovery, it's whole of the pharma or biopharma industry. Everywhere they use this information because they have lots of sharing of the data, healthcare data. Information is cheap for them, but understanding is expensive, was told by one professor. Why understanding is expensive? Take my example. I was not good at data analysis using the statistical software JMP previously, and they hired a data scientist who doesn't know anything about pharma or biopharma. When I talk to him, these are the cells. We maintain the pH, we maintain the temperature like this. After this processing condition, we get this output. Then he just closes his eyes for a moment and he says, I don't know what is that process. Because he doesn't have that background. When he talks about algorithm and all, I say, I really don't have any, I don't know what you're talking, Abhi, I say. So what if you guys have that, both the blended skills, I don't say you have to go so much deep into the artificial intelligence, but you at least to the level of understanding the data, what is happening in terms of clinical trials, what is happening in terms of manufacturing processes, what is happening in the R&D data. If you are able to analyze all of them in the pharma or biopharma, you will really have a bright future. There are lots of startup companies around the world who are working on only drug discovery using the artificial intelligence. It's just drug discovery. But there are a lot of other companies as well which are working on artificial intelligence in the manufacturing process, artificial intelligence in the patient related data, like this patient generated health data. This is becoming the next most important data. Incorporating patient reported outcomes into healthcare to engage patients and enhance care. Like I have given one example. The data was correlated with already existing drugs, which are very closely related to each other, and then they changed the drug to the woman and she got cured of the disease condition. One example of store using that data, like how do they store that? how pollen is happening in particular area every 12 hours, how temperature and humidity. And if you come to some asthma condition, what are the symptoms? What are the medicines? How many times? How many night wakings are there? Activity limitation. I don't know what exactly it is. I'm sorry. Is how many sleep alarms they had to have. What is happening in terms of body. So all the data is recorded and they see the health condition and they create a health dashboard. I heard some 4 years back some online dashboards of AP government. So similarly, it's going to be really uh, good to use artificial intelligence in drug discovery and patient related data. More interactions, role of data and analysis. How do they do that? The phenotypic drug discovery can be done. Like what is that exactly the phenotypic drug? Like which particular gene is responsible for that? Of course, they could be done even before also, even there was no artificial intelligence, but it takes really lot of time. And with the help of bioinformatics, they could do, but it was not involved with the algorithms. It goes in one particular flow direction only, even the bioinformatics and all. But artificially, it tries to loop everything, generate that algorithm, what is interacting with the what, how these remaining things are interacting with the next thing, and how the next thing is correlated with the next thing. So it creates a loop of algorithms and try to give the relation between the one disease to the another drug and to the another drug and to the another drug. That can be a good drug discovery. And they can predict the adverse event predictions. Like they do have a data of the patients, like suppose example 40 or 50 years old people, someone with cancer situation, they do have history of 50 years health data. These are the things they have passed through in the last 50 years. So the similar kind of observations, if they see in some other place in next 4 or 5 years, the first person got some cancerous issue. So the second person with the similar symptoms in the last 40 years, he might also get the cancerous issue. So then they can dig into that, genetical phenotypic evaluation and find, yes, there are something responsible for uh, future cancerous. So then they can start work on that. So combination therapy, that's precision medicine. So this all involves the data, healthcare data in drug discovery, healthcare data in precision medicine, healthcare data in the public health. There are a lot of sites which can give some information on artificial intelligence or drug discovery sources like PubChem, Swissport, and EMBL. Many of them could have been used by many people. So you'll have a lot of 96 million compounds relation, 260 million compound bioactivities. There's a lot of drugs, 10,000 to 56 drugs, 5 lakh 60,000 protein sequences. And you'll find a lot of nucleotide sequences, life sciences publications, and protein or structure related data, everything. Like example, crystal structure of ligand protein complex, predictive models. Already there are developed predictive models for many drugs. They can use them for the drug development. This is one example of drug discovery using artificial intelligence. There will be genes data, diseases data, there will be compounds data, and a lot of medical research, literature, etc., etc. Everything will go into the natural language processing using artificial intelligence, and and from all of them they create an entity attribute relation, like what is responsible for what is causing what is interacting with what. So all this information will be processed here through the data analysis and language processing. Then They'll be able to create potential target identifications. All it's not one place. This can be used in different stages, like interactions, associations, relevance in the therapeutic. Like one drug is already showed lot of thing, another closely related can show the relevance of the therapeutic activity. And clinical data, gene related associate, gene and disease associations. How one what exactly is that? There is a gene. That gene has that some symbol, and then for that particular gene, there is an associated disease, or change in the gene causes some particular disease, and it has a name. So these are given symbols and names, and there will be coding for that, and the data is stored, and similar situations will be immediately captured. This is one example. This is a drug, rosiglitazone. It works on particular gene which is linked with a lot of protein molecules, and these protein molecules with had some relation with the another gene structure which has a relation with all a lot of the drug. On these drugs, this this gene sequence has some transcriptions and translations, and those drugs have the impact on them, and that is having impact on myocardial infraction. This is a kind of heart situation. So they will be able to generate algorithms like this. It some other closely related thing can be used by some other drug, if not this one. That is how the drug is being discovered in the latest trends. Of course, they need some basic research, but it's making the things much quicker than what anyone can imagine. It's the same kind of example. Genes and disease associations, then get the data of the patients, 1 2 3, what kind of diseases are there, and what is the disease association symptoms, and everything is noted down. And there will be a projection model, what could be the problem, and what are the genes that are associated with them in the next another patient, and discover the disease phenotypic genes, and disease gene associations, and give the suitable drug to them. So exactly, either this machine learning is all nearly the same. Machine learning, natural language processing, everything is a part of artificial intelligence. How exactly they do that data comparison? How do they compare them? For example, if you see the molecule 1 and molecule 2 here, see the structures are very close, very close, except the position of some, I think, yeah, methyl group here on the benzene ring, right? So there will be they'll be giving a sequence to this, and the position of this will be varied, and position of this will be varied, and that is positions are given like this. And in one particular variation, they do have the output which is active. In another situation, it is inactive. So there will be more. It's not just, I'm not talking about only two models at this point of time. Imagine there are very, I mean, the CH3 can be here or here. So there are different lot of data which are very closely related, and the model will be created using this data, and then which is very closely related molecule, maybe some additional CH3 is there. So how it is going to impact? And most important features are X8 and X10, because these have shown the difference in whether it is active or not active. So that is how, and whether how much activity it could be having. So the prediction will be happen very much even before you try the drug in the lab scale. So it narrows down much further and further, and give you a idea to work on what are the minimum amount of hits that you can try to find the new drug. This is data science, which is one of the part of artificial intelligence. I particularly using JMP data science blended with biotechnological science. What it does is it gives you prediction, correlation, linear regression. What it does exactly? Like example, when I joined Ipsen, there is some 20 to 25. I'm not sure whether I am going beyond time. Can anyone say about it? It's getting over anyhow.
No.
Yeah, thank you. Uh, there was 20 years of the manufacturing data for one drug. So there were a lot of issues in the manufacturing. Every time one or the other batch is getting issues, there are variations because.
So excuse me,
Sorry.
sir.
Yeah,
Yeah, okay, please conclude in 5 minutes, sir.
yeah, sure. Thank you. So this data, you do the analysis and find out what is causing the issues. Then you see the correlation between the variations and output, which are having more impact on the output. Then you try to reduce the variations in that, and then that is how you control the drug production. This is also new trend apart from using the data science or artificial intelligence in the drug discovery. Yeah, these are the places where you can use this drug discovery, pharmacovigilance, adverse drug reactions, drug and drug interactions, personalized healthcare. But however, there are a lot of challenges to use this as well because it needs still for new drugs, it needs some amount of clinical trials and getting the data, even though it is cheaper, but it is difficult sometimes because there will be restrictions to provide the data. It's not easy. You need to get the consent from the people or from the organizations to get the data, so which is a legal issue. There are a lot of companies working on the using the artificial intelligence in the drug development at different levels, genomics, cellular, and also molecular therapy. Uh, these are the major companies they are working on AI, using AI in the drug discovery. So we one among them. It's the same. Talks about the post-marketing analysis, patient relevant evidence to provide the suitable drug, and also multiple across the multiple databases, how it can be correlated for the drug discovery. This is also an example of how the machine learning is working on how they get the data and data extraction and automated scoring. This is one example of how the data is read by the artificial intelligence in the drug discovery. If you see aspirin molecule is one way of reading that. They give the sequence like this in the programming. If they if they get this, that means aspirin. Similarly, this is actually the structure of the molecule. So what are the molecules in what order it is there in the same way they give the sequence. Similarly, all the other drug molecules like any other proteins or then they will be given the sequences and they will correlate. There is another one fingerprint encoding and molecular descriptors also like each and every type of if it is methyl group one code will be given. If it is benzene ring one code will be given. So and the order of the codes will be also is important in this. There are a lot of areas where artificial intelligence is being used in terms of drug discovery and development. There are real world examples. They identified a cancer treatment compound recently, and also they identified one protein inhibitor through the artificial intelligence itself, and they identified uh, therapeutic drug for the Alzheimer's disease also. And there is a novel antibiotic discovery. And recently there is this work still going on in the COVID-19, and but there is one example which is remdesivir. for the COVID-19. They used initially this artificial intelligence to find this can be a potential drug which is already in the market. So what at the present we all have this pharmaceutical and medical experience. We all know some regulatory requirements. We all know some cross-functional team collaboration. But what is needed in the future is blending of the computer skills with the scientific skills. It's not too far. It is just within 4 or 5 years probably. Everyone gets updated. If you are coming out into the market, you have to be equipped with all of them. Thank you. Thank you so much. And it's nice to interact with all of you. Any questions?
No, sir. Thank you, sir. Thank you for your presentation and valid information.
Thank
And
you.
once
Thank you
again,
for
thanks
giving
for
me this opportunity.
thank you for your acceptance when we are asked about the to participate in FDP as a resource person. Thank you, sir.
Thank you. Thank you. Thank you so much.
Thank you, sir. No,
And
thank you.
wish you all the best in your future endeavors. Thank you. Have a good time.
Professor.